Hello everyone, my name is Anastasia and today I'm going to tell you about the paper that was published in 2016 in Cell Journal. The paper title is Precision Tumor Recognition by T Cells with Combinatorial Antigen Sensing Circuits. This article was cited 219 times. Immunotherapy based on T cells carrying chimeric antigen receptors or CARs represents a promising approach in cancer treatment. And currently, clinical trials are conducted in many countries all over the world. To date, the FDA approved two CAR T cells therapies to treat pediatric B-cell acute lymphoblastic leukemia and B-cell lymphoma in adults. During the therapy, T cells are purified from blood drawn from the person and then modified by virus that inserts a CAR gene into the cell's genome. The modified T cells expressing chimeric antigen receptors on their surface are reintroduced to the patient. These CAR T cells then recognize and kill tumor cells. The receptor that recognizes tumor cells is a chimera. The intracellular part represents a signaling domain of the T cell receptor complex, whereas the extracellular part is an antigen recognition domain of an antibody. The binding between CAR and its antigen on tumor cell surface triggers a signal transduction cascade through signaling domain and then activates T cells to kill the target directly or through other components of the immune system. Previous generations of CAR T cells expressed one chimeric antigen receptor. The major limitation for these T cells is their interaction not only with tumor cells, but also with normal tissue cells that could express the antigen. So the question is how to make T cells that could reliably discriminate normal tissue from disease. To increase the specificity of the CAR T cells, dual targeting approaches have been developed. In this case, two chimeric antigen receptors are expressed by T cell. However, partial signaling through one of the receptors can generate sufficient T cell activity and cause off-target tissue damage. In this study, scientists developed the combinatorial antigen system containing synthetic notch receptor and inducible CAR receptor. In this case, two antigens are required for T cell activation. Now, let's have a look at the design of combinatorial antigen receptors. To create the first antigen receptor, scientists modified the wild-type notch receptor. They selected the notch pathway because of its very direct and simple mechanism of signal transduction. Engagement of the notch receptor with its ligand, delta family proteins, leads to cleavage of intracellular domain and its migration to the nucleus, where it activates the expression of target genes. Synthetic notch receptor, or synNotch, was engineered by changing the extracellular and intracellular domains. Upon interaction with an antigen, the cytosolic domain, which represents a transcription factor, migrates to the nucleus and induces the expression of CAR gene. However, if the target cell had only one antigen, the activation of T cells does not occur. The CAR mediates T cell activation only if the second antigen is present. In this case, activated T cells induce the target cell death. This strategy has the potential to overcome off-target reactions that occur with conventional CAR T cells. After that, scientists checked the expression of synNotch-driven CAR in human primary T cells. The synNotch receptor was designed to interact with GFP protein, whereas the CAR targeted CD19 molecule, which is a gold standard in the field of immunotherapy. These T cells were exposed to myelogenous leukemia target cells, expressing CD19 only, GFP only, or both GFP and CD19. T cells were activated only when exposed to cells expressing both CD19 and GFP, and this activation of T cells resulted in target cell death. Then scientists tested whether the synNotch CAR T cells could selectively clear a dual antigen tumor in vivo. They set up a bilateral tumor model with malignant leukemia cells. On the left flank, they injected tumor cells expressing CD19, whereas on the right flank, tumor cells expressing both GFP and CD19. After 4 days of implantation period, they injected engineered T cells and monitored tumor growth for 20 days. CARs were expressed only in T cells interacting with dual antigen tumor, leading to activation of T cells and tumor cell death. Activated T cells selectively clear the dual antigen tumor on the right while leaving the single antigen tumor on the left. These synNotch T cells displayed high and reproducible discriminatory action against two tumors present in the same animal. To conclude, the use of synNotch receptors has the great potential to improve the safety and effectiveness of T cell therapy. Engineered synNotch CAR T cells are activated only by dual antigen recognition, providing highly controlled functioning with no observable off-target toxicity. Moreover, these synNotch CAR circuits could expand the set of antigens that can be targeted by immunotherapy.